discuss about pairwise sequence alignment using BLAST. Pairwise sequence alignment, also called pattern matching, is used to identify regions of similarity that may indicate functional, structural, and/or evolutionary relationship between two biological sequences, can be a protein or nucleic acid. We find three important technical terms in pattern matching: number one, match; number two, mismatch; and number three, gap. If you compare two sequences, sequence 1 and sequence 2, if you find the similarities, we call that as a match. And if you find dissimilarities, we call it as a mismatch. And if you find a missing character, we call that as a gap. So types of pairwise sequence element we can classify as dot matrix method, Ford or K-tuple method, and dynamic programming method. So these are the corresponding softwares for pairwise sequence alignment. Also, we can classify it as local or global alignment. So BLAST comes under local alignment, and if you see the expansion of BLAST, basic local alignment search tool. So these tools are present in almost all the public domain databases. And if you try to search databases versus BLAST, so we generally search a query sequence versus BLAST. If you try to search a nucleic acid versus nucleic acid, you can use BLAST and protocol. And if you search protein versus protein database, you can use BLAST-P. And if you search for mRNA versus mRNA, we can go for T-BLAST-X. And if you search mRNA or conceptual protein translations versus protein, we can use BLAST-X, and if you search protein query versus mRNA, we can use T-BLAST-N. And here in NCBI BLAST, if you want to search a protein related sequences, okay, you can type the corresponding protein sequence in FASTA format or accession number or GI in the specific region. And in this region, if you click BLAST, you can run the BLAST. Finally, you can get an output like a graphical output as shown in this. The alignment based on alignment scores, you can find the kind of hits arising in different colors. And in this slide, you can find also the text. The output can also be available in the form of a text. And these are the hyperlink to the sequences. And this is the bit score and e-value, which you can find as a text kind of an output arising from class. Also, you can find in this slide the e-value and bit score. Now we can take a problem statement to how to retrieve a human insulin using class P. So now we can take you to the browser. So we can search for NCBI. So NCBI is National Center for Biotechnology Information, which is hosted by NIH, National Institute of Health. Now we have to search a Homo sapiens protein, and we have to search a similar sequences of Homo sapiens protein. If you take that kind of a problem statement, okay. So I initially selected a protein database and searching for a insulin protein. So now we are getting so many kind of proteins. Now I have to check the specific Homo sapiens insulin. So I am going to click here. So after clicking in Homo sapiens insulin, you are getting the proteins. Now I'm going to search in FASTA format. So whatever insulin I have searched in FASTA format, I'm going to click Run Blast, which is present in the right hand side. So I can find regions of similarity. You find the accession number now, which you're going to search in Blast P. Simply click Blast. If you click Blast, okay, you find the form of output like this. So you can find a graphical output and descriptions like e-value query coverage is mentioned here. So thank you.